we move to environmental lipidomics with our next speaker, that is Professor Cinta Porte from the Spanish National Research Council. So Cinta, if you're ready.
Okay, thank you. Can you see my screen?
Yeah, we can see it now.
Yeah, okay, thank you. Just rinse this. Okay, so thank you very much for the invitation to participate in this workshop. I think it's really interesting to gather different omic perspectives. And yeah, my talk is about lipidomics, that is something slightly different, and mainly about environmental lipidomics in the sense that is like an emerging field in the omics section that investigates how environmental factors influence lipid composition and lipid dynamics in cells and organisms. And very, very briefly, I would like just to introduce the lipids. I mean, because lipids, we all know about lipids, but this is a very, very heterogeneous group of compounds. There are more than 10,000 lipids, 10,000 different molecules they have very, very diverse structures, different characteristics, and what is more important, different functions. And the only common characteristic is the fact that there are water-insoluble compounds, because, for instance, regarding functions, they have a very important role in membranes. They work as structural lipids, as building blocks of membranes in the cell. They also serve as energy storage, and there are many of them. There are bioactive molecules. For instance, they are involved in cell signaling, in apoptosis processes and inflammation and so on. So from this perspective, are really a very interesting group with very different functions and also very dynamic, because they respond to external stimuli, for instance, changes in temperature or nutrition, aging, reproduction, and also exposure to pollutants. And this is something that is very poorly investigated, even though we know that lipids play a key role storing contaminants and so on. So another aspect to consider is the fact, for instance, that lipidomics has been investigated or used as a tool very much in biomedicine, for instance, in cancer research. However, when it for aquatic organisms, in fact, in aquatic toxicology, we have not really used this tool. So, having said so, I would like just to introduce the outline of the talk. I divided the talk in 4 sections. I will try to show you how exposure to contaminants, so mainly to plastic additives, because I mean, just to focus in a group of chemicals, how the exposure to these plastic additives is able to modulate the lipidome. And in the first part, we will see some examples of exposure to single compounds. After that, we will go to how the mixture of these plastic additives can or cannot modulate the lipidome of the cells. We will also consider the particles, so micro and nanoplastics. Do they affect the lipidome and how? And finally, I will briefly comment a field study where we were taking like a fish muscle biopsy, and these fish are exposed to a mixture of unknown pollutants, but they were taken in areas that are polluted mainly by urban and industrial wastewater. So most of the time, the models I will be talking about will be fish liver cells, PLHC1 and zebrafish liver cells, and in the last part is just a piece of fish muscle tissue. Also, before starting, I would like to very briefly introduce mass spectrometry, because nowadays is really the development of mass spectrometry that has allowed us to really monitor and investigate the lipidome of different models. And in our group, we've been mainly using these two techniques that appear in the slide. The first one is just the direct infusion of the lipid extract in the MS analyzer, is what is called shotgun lipidomics. And this technique has the advantage that it's very simple. It's very fast. In 2 minutes, 3 minutes, you have the lipidome of your model. And there is no need to operate the liquid chromatography parallel to the mass spectrometer. It is also very useful in terms of quantification, in the sense that ion suppression and matrix effects are the same for the analytes and for the internal standard, so your quantification is very good. But it has the disadvantage that there is a strong ion suppression, so we can only measure the most abundant lipids. So from time to time, we are also using another approach that is liquid chromatography coupled to mass spectrometry, and with an analyzer like an Orbitrap or a Q-TOF analyzer. Regarding our work with cells, I just want to show you very briefly the type of methodology, just to show that this is pretty straightforward. We can culture the cells or expose the cells in a format like the one in the slide. It's a 24-well format. This is fine. So you expose the cells for 24 hours, and after that you have to remove the culture medium, and this amount of cells that it remains in the well is enough for analyzing the lipids. So we detach the cells, we extract the lipids with a mixture of organic solvents, and we can really just this extract directly inject it into the LC-MS instrument. Sorry. Before continuing, a brief introduction about plastic additives, that are the group of chemicals we are going to talk about. These are chemicals that are used to improve the properties of the polymers. And it is, there is a very large, there are many compounds that are nowadays used as plastic additives. In fact, there are more than 10,000 chemicals classified as additives, and you can see in the slide that a very important group are the plasticizers. Many of them are phthalates. We have also fillers, flame retardants, antioxidants, heat stabilizers, colorants, and so on. So a very large number of chemicals, and a quarter of them, they are classified nowadays as compounds of potential concern, either because they have been classified as persistent, or they have shown bioaccumulative properties, or they have been shown toxicity. So what is the problem of these chemicals? Well, these chemicals are added to the polymer, and they are not chemically bound to the polymer. So if the plastic ends up in the aquatic environment, for instance, or the plastic enters in contact with food or liquids, they can leach out, migrate, and contaminate. And nowadays, there are really ubiquitous contaminants. And some of them have been classified as opisogens, endocrine disruptors, and so on. But we wanted to see what is the effect at the cellular level in terms of the lipidome. Are they really able to modulate the lipidome? And this is an example of some of the studies we've been carried out. We were exposing PLHC1 cells to phthalates, to different concentrations of phthalates, just for 24 hours. And after that, we measured the lipids, and you can see here the volcano plots. And in red, each dot is a lipid, and the ones highlighted in red correspond to the ones that have been significantly increased or significantly decreased in exposed cells. And you can see here the results for two exposure experiments. One is 5 micromolar diethylexyl phthalate, and the other one on the right, 20 micromolar dibutyl phthalates. The red color means so 5 micromolar or 20 micromolar are the nominal concentrations, but in fact, these are pretty hydrophobic pollutants. So to improve the sensitivity of the method and also the reliability of the results, what we do is to measure the real concentration of these contaminants in the exposure medium. So as you can see, the 5 micromolar DHP is the theoretical concentration, but the experimental concentration was 1 micromolar, and for DBP is the same. The experimental concentration was not 20, but 3 micromolar. And as I said, both compounds, you can see they induce the accumulation or they increase the accumulation of some lipids inside the cells, and these lipids are mainly triglycerides. So they have in these fish liver cells, they are having a kind of obesogenic effect. And in fact, this is occurring at concentrations of phthalates that have been described in fish, for instance, about 400 or 4 to 800, 900 nanograms per gram. This is regarding phthalates. This is another example for bisphenol F. Bisphenol F is one of these bisphenols that are used nowadays in replacement of bisphenol A. So 24 hours exposure of PLHC1 cells, and this is the volcano plot. And you can see here that after exposure to 20 micromolar BPF, you can see on the left part side of the screen that a number of lipids were depleted, and mainly a group of them were triglycerides. So in lipidomics, it's always difficult. Sometimes the interpretation of the results, and it gives you a lot of work and a lot of new experiments to be carried out. So bisphenol F is you can see in the slide that bisphenol F it induces in fish liver cells, it induces the accumulation of or the generation of reactive oxygen species. And this is our hypothesis is that this induction of reactive oxygen species will generate oxidative stress, and is very likely that it will oxidize the phosphatidylcholines and other lipids. So it has been reported already that triglycerides, the hydrolysis of triglycerides is a way to liberate new fatty acids that can be used to replace and synthesize new phosphatidylcholines. If we increase the concentration, that is already pretty high, 20 micromolar, but if we move to 100 micromolar, there are more and more changes that are being, more changes in lipids that can be observed. For instance, you can see this decrease on certain phosphatidylcholines, plasmalogens, PCPs, PCOs, and so on that I will explain later. But obviously, the higher the exposure, more and more pathways of lipid synthesis and lipid metabolism are affected. So this is about single compounds. We did work with several other contaminants, and well, some of them we have different like lipidic fingerprints. But finally, we decided to work with mixtures in the sense that aquatic organisms are never exposed to a single phthalate or a single plastic additives. So in the slide, you have a mixture of 10 plastic additives that are widely reported in the aquatic environment. It contains phthalates, bisphenols, alkyl phenols like octyl and nonyl phenol, triclosan, that is an antimicrobial, and the two last ones are two flame retardants. And they were tested at a concentration of 1 micromolar each one. So we have a mixture of 10 micromolar, 1 micromolar each compound. We exposed the cells for 24 hours, and what we observed was a really good separation between the lipidome of control cells and exposed cells. And you can see here the volcano plot, and well, a number of lipids were upregulated. These were mainly phosphatidylcholines and phosphatidylethanolamines containing palmitic acid, fatty acid 16-0. And this was quite surprising, the effect of the mixture. But it's also interesting to see that this is the type of lipids that are modulated are those containing palmitic acid, and palmitic acid is known to induce inflammatory responses in the liver. And also, it's also important to consider the fact that this means in terms of membranes a decrease in the levels of lipid unsaturation, and this may at the end alter membrane functions like fluidity. But I mean, this is just an hypothesis that we observe these changes also in membrane fluidity, or have been described for guanylyl phenols that were part of the mixture. And well, this is about this part of the talk goes about what is the effect. We were wondering what is the effect of plastic particles, because plastic particles are really plastic micro and nanoplastics are really a highly diverse class of contaminants, because the size is very different. They span over 6 orders of magnitude in size, very different shapes, different composition. Apart from the plastic additive, we have residual monomers, we have the polymer, and so on. So this is an issue that is very difficult to address in the aquatic environment, and we just decided to go for the simplest cases scenario. So we were working with just polyester plastic micro and nanoparticles from 1 micrometer and 50 nanometers. And we were exposing zebrafish liver cells for 24 hours at different concentrations. And after that, we will just measuring the different endpoints like changes in cell viability, reproduction of reactive oxygen species. So imagine, as you can see here, that we would see that some of the microplastics really enter into the cell, and also lipidomics. And for instance, regarding changes in cell viability, we can see that microplastics did not change cell viability in our system after 24 hours exposure to different concentrations, that they go from 2 to 135 micrograms per milliliter. However, we see a certain decrease in cell viability, very low, but about 10% decrease, 15% decrease for decrease of cell viability for nanoplastics. And both of them, micro and nanoplastics, induce regeneration, but at that really very low level. What is interesting to know is that, well, regarding lipidomic responses, we were expecting lipidomic responses for all of them, for micro and for nanoplastics, because microplastics enter in the cell, so there is a reorganization of cell membranes and some. But in fact, it's interesting to know that for microplastics, if you look at when we look at the lipidome of the cells and the exposed cells and control cells, it was only possible to differentiate the lipidome between the control cells and those exposed to the highest concentration, that it was pretty high, 135 micrograms per milliliter. However, for nanoplastic, and you can see in the bottom, it was possible to see really a discrimination between the lipidome of all the different exposure groups. You can see in red the control, in green the lowest concentration. That was also almost possible to fully discriminate the lipidome and a kind of dose response change in the lipidome. This can be seen also here in terms of volcano plots. On the left-hand side, you can see the plot for microplastics. Microplastics are the highest concentration. What it happened, it induced a decrease of lipid synthesis, but mainly polyunsaturated phospholipids, among also some cholesterol esters and phosphatidylinositol and some other groups. But it's very interesting to see this decrease, this generalized decrease in lipid synthesis. But this is a concentration that is, I mean, there is not a lot of information, but we think that is pretty high and not of environmental relevance. In contrast, for nanoplastic, the response was much more complex, and what we can see is an accumulation on the right-hand side of many different lipids. Let's see this response to nanoplastics. 3 different concentrations of nanoplastics from 2.6 to 130 micrograms per milliliter. And you can see in the upper part that exposure to nanoplastics lead to an upregulation of these PCOs that we will be talking about later on. And the higher the concentration, the more lipids were affected. Sometimes it's really difficult when you have identified 200, 300, or even more lipids to try to put these lipids into a context to try to interpret the results. So we are nowadays using this pathway analysis, and this allows you to simplify or to put in order some of these findings. And these are the active lipid reactions observed for nanoplastic exposure, polyestyrene nanoplastics of 50 nanometers. The low concentration is close to what sometimes has been reported as an environmental concentration. And these are the active reaction is just this conversion of phosphatidylserine to phosphatidylethanolamine. But what we can see is when we increase the concentration, we can observe an activation of this lysophosphatidylethanolamine pathway. So what is happening at the medium concentration, this reaction is activated, but also the hydrolysis of phosphatidylethanolamines to lysophosphatidylethanolamines. And the activation of this pathway has been linked to lipid droplet formation. And it's very interesting to see that when this occurs, it also activated this pathway here, the synthesis of triglycerides. And also the same pathways are activated at the highest concentration. But I mean, with a higher activation, with a higher significance of this activation. The bottom part, we can see that is the activation of ceramides and of the ceramide pathway, mediated apoptosis pathway. And you can see that also this effect increased with concentration. For instance, no effect at the lowest concentration, where at the medium concentration, we can observe the accumulation of this ceramide and this one, and at the highest concentration, we can see the accumulations of these 2 ceramides that are linked to apoptosis. So we move now to the last part of the talk. I mean, that is a field study where we were taking samples of fish from a Mediterranean river located near the area of Barcelona. We took samples from 6 sites, 3 reference sites that were located in the upper part of the river. It looks something like this, not a lot of water. And 3 sites that they were located downstream. And we know that, well, this is a quite urbanized area, so we know that this river is affected by the output of wastewater treatment plants and some other effluents. And these were the two species that were investigated, Barbus meridionalis and Squalius laetanus. They were fished there, and we took a small piece of muscle tissue. In fact, we were thinking about the possibility of just taking a small biopsy of the muscle tissue. And this is a cluster analysis, and this is for this species, the Barbus meridionalis, that is the most benthic species, and it lives in contact with the sediment, and is highly exposed in the polluted area, it's highly exposed to contaminants. And we could see that there is a quite good, cluster of the polluted sites and the reference site clustered very well together separately. And even for the reference sites, it was possible the different areas clustered together. I mean, in fact, in each site, we have 3 samples, and each sample is a pool of 3 different individuals. So you can see it was possible to see that in the lipidome of the muscle of fish living in these areas was very, very well separated between polluted and reference sites. For this one is still polluted and separate, and reference sites were well separated, but the clustering between the same areas is a bit more difficult. So just let's analyze some of the changes that we observe. For instance, if we measure phosphatidylcholines in the muscle tissue, in both species, Barbus meridionalis and Squalius, we can see that in polluted sites there is a certain decrease here for Barbus, and is much more evident for the other species. A strong decrease in these species that are phosphatidylcholines with a high number of double bonds, and therefore these are the phosphatidylcholines that are more susceptible to oxidation. So we can see, as I said, an oxidation of these phosphatidylcholines, mainly in this one, and also the generation of these hydrolysis products that are lysophosphatidylcholines. And this is more evident in one species than in the other. Also very interesting, these results here, that is an increase of PCOs. These are ether-linked phosphatidylcholines. They have a structure like this one, and we have plasmanyl lipids with no double bond, and plasmanyl lipids of plasmalogens with this double bond. For PCOs or plasmanyl lipids, we really don't know at this moment what is the function of these lipids, but they have been described high levels of these PCOs. For instance, we observe after nanoplastic exposure, or they have been described in human tumor tissues. And you can see how one of the species, the Barbus meridionalis, in the polluted sites, it always shows an increase in PCOs, and also the other one, although not so evident. And also, it is important to see that these ones, plasmalogen, plasmanyl lipids, or plasmalogens, or PCPs, the function of these lipids is known, and they work as endogenous antioxidants. They are very susceptible to oxidation. So in an area with high levels of reactive oxygen species, or when the organism is exposed to this kind of pollutants and generate reactive oxygen species and oxidative stress, what it happens is that these lipids oxidize and protect from oxidation the phosphatidylcholines. And you can see that for this species, there is this decrease in the polluted site of PCPs, whereas for the other one, there is no change. And it's interesting if we go back that this one that had a decreased level of PCPs, somehow they have protected species from oxidation, whether the other one is really suffering the oxidation of this, of phosphatidylcholines. And yeah, also a general trend that it has been described in, or we observe in this study, and we observe also in other studies, is a dysregulation of cholesterol metabolism. And we can see that both species show an increase in cholesterol esters in the polluted sites. And in fact, just to summarize some of the results, we can observe in our lipidomic studies that there are always some common alterations that they have the potential to be investigated as potential biomarkers of exposure. For instance, this enrichment of triglycerides and cholesterol esters, and the enrichment of PCOs, that we don't really know the function of these lipids, but is always a threat that is pretty common in some exposures, and the increase of lysophosphatidylcholines. Also, in conditions of oxidative stress, it's also possible to see a depletion of highly unsaturated phosphatidylcholines and phosphatidylethanolamines, and significant changes in the metabolism of ceramides and sphingomyelins. And we think that while the application of lipidomics in toxicology studies can be very useful to help to obtain a signature of how pollutants act, the mode of action of pollutants. It's easy to see if they generate oxidative stress, inflammation, or some kind of metabolic disturbance. And yeah, we think that really lipidomics is really a good tool, or a promising tool for together with NAMs and non-invasive monitoring strategies to apply in the future. And it has a lot of potential. And with that, I would like just to thank you for your attention, and thank my co-workers that have done a very excellent work in this field. Thank you.
Thank you so much.